Welcome back for another deep dive. You guys sent in some really interesting research on protothecosis in dogs. We're going to be taking a look at two case studies from the Journal of Comparative Pathology. These cases really give us a unique look into this rare disease. And just so we're all on the same page, protothecosis is an infection. And it's caused by a type of algae called Prototheca. I'm sure some of you are thinking, wait, algae? That's something you find in a pond, not in our dogs, right? So let's unpack this a little.
Protothecosis is a really good example of the One Health concept. It can affect dogs, humans, and other animals too. And it's not what we consider a classic zoonotic disease, meaning it's not something that easily jumps from animals to humans. But it does really highlight how interconnected the health of animals and humans actually is.
Yeah, it really emphasizes that link, doesn't it? So in these two cases, we have a Pekingese and a German Shepherd. And both of them were dealing with chronic bloody diarrhea and sudden blindness. I can only imagine how worried the owners must have been.
Oh, absolutely. And on top of that, diagnosing protothecosis, it can be a real challenge even for experienced veterinarians. They use a lot of different tools to try and figure it out. Things like cytology, histopathology, cultures, and even PCR. But the tricky part is that the symptoms, they can look a lot like other conditions, so it can be tough to pinpoint exactly what's going on.
So let's dive into the case studies themselves. The research paper goes into some pretty dramatic findings from the post-mortem exams. What really jumped out at you?
The macroscopic lesions, they were really striking. There were these white nodules on the heart, skeletal muscles, and even lining the intestines. And the intestines themselves were thickened and discolored, which is a classic sign of severe inflammation.
Wow, that paints a pretty vivid picture. It really shows you just how serious this infection could be. What about the microscopic findings? What did those reveal?
When they looked under the microscope, they could actually see the Prototheca bovis, which is the algae causing all the trouble, in various organs. It's almost like a microscopic invasion. And they even found it within the blood vessels, which suggests that it can actually spread throughout the entire body.
So that helps explain why there was so much damage and why those symptoms like bloody diarrhea and blindness were so severe. But the burning question here is, how do dogs get protothecosis in the first place?
Well, the most likely way seems to be through ingestion. Dogs might come across this algae in contaminated environments, and then it gets into their system through the intestines.
So something like a contaminated water source could be the culprit.
Exactly. But here's where things get really interesting. Not every dog who's exposed to Prototheca actually gets sick. Genetics probably plays a big role, especially when it comes to the immune response. Some dogs might have a genetic predisposition that makes them more susceptible to this infection.
That makes a lot of sense. So it's not just about being exposed, it's also about how each dog's immune system reacts to that exposure. Did these two dogs in the case studies show any signs of a weakened immune system?
That's the fascinating part. They didn't, which actually makes it even more complex. It really highlights just how much we still don't know about protothecosis and what causes these really severe cases to develop.
Wow, so we have this algae that enters through the intestines. And it can spread through the bloodstream, causing all these problems. But treating it, that's going to be a real challenge, right?
Absolutely. Treating protothecosis, especially when it spreads throughout the body, is extremely difficult. The algae is often resistant to the antimicrobial medications we typically use.
So what options do veterinarians have when they're facing this diagnosis? What can they do?
Unfortunately, finding effective antifungal drugs for animals is a real struggle. And treating systemic protothecosis, it's usually unsuccessful.
That's pretty discouraging, especially since the research mentioned that the disseminated form, that's the most common way it shows up in dogs. Is that right?
Yeah, that's accurate. And just like we saw in these cases, it usually starts with diarrhea, and it's often resistant to the typical treatments.
The authors of the studies suggest that the infection probably starts with ingestion and that the colonic mucosa is like the entry point. But how does it actually spread from there? Like, how does it get beyond the intestines?
Well, the theory is that once you have that initial lesion in the intestines, it kind of creates an opening for the Prototheca to get into the bloodstream, possibly through lymphatic drainage and the movement of white blood cells. And the fact that they found lymphatic and vascular thrombosis in the intestines, that really supports this idea.
So it gets into the bloodstream, that makes sense. But why don't all dogs who are exposed to this algae end up with the disseminated form? What makes these cases so severe?
That's the million-dollar question. We know that immunosuppression is a factor in humans, but there's not enough evidence to say the same for dogs. Researchers think that genetic factors might be more important than just being exposed to the algae, things like innate immunity and the ability of cells to actually engulf and destroy pathogens.
So some dogs might be genetically predisposed to developing this disseminated form of protothecosis. It's not just about being exposed, but about how susceptible an individual dog is.
Exactly. And this is backed up by the fact that protothecosis isn't really common in dogs, even in areas where there's a lot of environmental contamination from bovine mastitis caused by Prototheca.
So even in places where the algae is everywhere, not all dogs are getting sick. It's amazing how much individual factors seem to matter. Both of the dogs in these case studies suddenly went blind. What causes that? How does this algae affect their vision?
The thinking is that the Prototheca sporangia travel to the eye through the bloodstream, and then they affect the vascular structures, which leads to something called exudative chorioretinitis. Basically, it's inflammation and fluid buildup in the back of the eye. And all that inflammation and necrosis eventually causes the retina to detach, and that's what leads to blindness.
So it's not just the intestines this algae can affect all sorts of organs, including the eyes. The researchers describe the lesions as looking like myriads of brown millimeter-sized nodules. Did they find those nodules in other organs besides the heart, kidneys, and intestines?
Yes, they also found them in skeletal muscle. And there are reports of Prototheca affecting the eyes and the central nervous system, leading to blindness and neurological problems like paraparesis, which is weakness or paralysis in the hind limbs.
Wow, it sounds like this algae can have a pretty wide range of effects, depending on where it spreads in the body. But these dogs in the case studies didn't show any neurological signs, right?
That's right. They didn't have any neurological symptoms, and no lesions were found in the brain or spinal cord. Interestingly enough, there's actually a reported case of protothecosis in a Labrador retriever, where the only lesions were in the central nervous system. This just shows how much the presentation can vary from case to case, and it really underscores how much we still have to learn about how this disease manifests.
It's incredible how different it can look from one dog to the next. When the researchers described the microscopic lesions, they mentioned granulomas, neutrophilic infiltrates, and sporangia. Did they see a consistent pattern across all the affected tissues?
Actually, no. While some areas had that classic inflammatory response with granulomas and infiltrates, other areas just had the Prototheca sporangia without any inflammation around them. This suggests that the way the host responds to the algae can vary a lot.
That's a really important observation. The study emphasizes the importance of using special stains to see the sporangia and sporangiospores. Stains like PAS and Grocot's methenamine silver. Why are those stains so crucial for making the diagnosis? What makes them so important in these cases?
Well, these stains basically help to highlight the Prototheca organisms, so they're easier to spot under the microscope. And this is especially important because Prototheca can be mistaken for other fungi or algae, like chlorella, for instance.
So being able to tell the difference between Prototheca and chlorella is important for getting the right diagnosis.
Oh, absolutely. They look really similar under the microscope. But chlorella has a green pigment that you can actually see even in formalin fixed tissues. And it also has starch granules in its cytoplasm that can be stained with PAS. However, the most reliable way to tell them apart is to use transmission electron microscopy.
And in these cases, molecular analysis was used to confirm that it was Prototheca bovis. Is that species commonly found in dogs with protothecosis? Is it the usual suspect, so to speak?
Yes, Prototheca bovis, which used to be called Prototheca zopfi genotype 2, that's the ones most often identified in dogs, Prototheca wickerhamii, and Prototheca ciferri, which was formerly known as Prototheca zopfi genotype 1. Those are less common.
The authors highlight that their study is really important because it provides such detailed descriptions of the macroscopic and microscopic lesions associated with protothecosis. Why is that so significant? What makes that kind of detailed description so valuable?
You see, protothecosis is relatively rare in dogs, and a lot of the earlier studies focus mainly on the clinical signs, or on identifying the organism using molecular techniques. But this study actually gives us valuable information about what the disease looks like at a cellular level, which can help veterinarians understand and diagnose protothecosis better.
So this research gives us a more complete picture of protothecosis in dogs. It's amazing how much we've learned from just these two cases. What are the main takeaways for vets? What should they keep in mind when they're dealing with a potential case of protothecosis?
Well, first of all, protothecosis should definitely be on their radar as a possible diagnosis in dogs with chronic bloody diarrhea that isn't responding to treatment, especially if the dog is also showing signs of vision problems. Second, using those special stains like PAS and Grocot's methenamine silver, that can help them actually see the organism and confirm the diagnosis. And finally, even though treatment is tough, just being aware of this rare disease is crucial for giving those dogs the right care and management.
This deep dive has been incredibly insightful. Protothecosis is a truly complex and fascinating disease. What's one thing you want our listeners to take away from our discussion?
Well, I think the biggest takeaway here is that even though we've been focusing on dogs, it really makes you think about what this means for other species too. Even humans, you know. That's something to really consider.
You're absolutely right. It really highlights that connection between animal health and human health. We've covered a lot of ground today, from the pathology and diagnosis to the challenges of treating protothecosis. But before we wrap up, I wanted to go back to something you mentioned earlier about using PCR for diagnosis. Can you break down how PCR works and why it's so valuable for confirming protothecosis? What makes it such a powerful tool?
Sure. PCR stands for Polymerase chain reaction, and it's a technique that allows us to amplify specific DNA sequences, so basically we can make them detectable. In the case of protothecosis, researchers can take DNA from tissue samples and use these things called primers that specifically target the DNA of Prototheca. If Prototheca DNA is present, the PCR process will create millions of copies of that specific sequence, and then this amplified DNA can be visualized, which confirms that Prototheca is there. PCR can even tell us which species of Prototheca is present, like we saw in these case studies.
So it's incredibly sensitive and specific. It's almost like finding a needle in a haystack, but for DNA.
Yeah.
It can pinpoint the exact culprit.
Exactly. And in cases where the traditional methods like cytology or culture aren't giving us a clear answer, PCR can provide that definitive diagnosis. It's really a game changer in those tricky situations.
That's remarkable. It's amazing how technology like PCR is changing the landscape of veterinary diagnostics. This deep dive has been a real journey into the world of protothecosis, a disease that reminds us of the importance of continuous learning and staying informed. We've uncovered so much about this rare infection, from its unusual origin in algae to its devastating effects on dogs. It's a clear reminder that animal health and human health are interconnected, and that we need to keep researching to improve our understanding and treatment options for protothecosis. From Vet Neurojar, keep those minds-inspired hearts light and tails wagging.